So our next speaker is Chiara Cantos, who interned at the Norris Center, and her mentor was Andy Koloskowski, but I know that there was a whole team involved because there's a number of folks who interned there this summer. Let's see. So the title of her talk is Past and Present Pollinator Community Composition in Californian Coastal Habitats. Okay, take it away, Chiara.
Okay. Hello, everyone. My name is Chiara Cantos, and this summer I interned with the Kenneth Norris Center of Natural History. During my time there, we studied past and present pollinator community composition in Santa Cruz County. So why are bees and pollinators so important? Pollination is key to ecosystem function, allowing plants to flower, produce food, and create more offspring. Because we live in an area that prioritizes natural beauty and agriculture, pollination is vital to our existence. However, it is a fragile system that can be affected by environmental changes over time. Examples include the presence of climate change and the effects of drought, habitat availability as urbanization and habitat fragmentation increases, and overall plant community composition. As a result, pollinator abundance and diversity decline over time. This leads to a cycle that can harm agricultural interests of Santa Cruz County, as well as Californian endemism and biodiversity. So for my research question, I focused on two main questions. The first was the influence of habitat on pollinator community composition. The two main habitats we studied and differentiated were coastal dune and coastal scrub. Coastal dunes are sparse in vegetation due to the harsh environmental factors, holding few plants that can sustain the sand, wind, saltwater, and cold temperatures. Coastal scrub, on the other hand, has greater plant species richness due to the more tolerable environment compared to the dunes, such as warmer temperatures, lower wind speed, and lower salinity. My second question was if there was a difference between the same habitats over time. In addition to the data we collected this summer, we had access to naturalist Randall Morgan's pollinator collection from the 1990s, graciously curated by the Norris Center. This allowed for a comparison between the 1990s habitats and the present day habitats. So the past is on the left and the present is on the right. To conduct this research, we surveyed pollinating bees and flies at 4 sites in Santa Cruz County from June to August 2021. We ran 4 transects monthly, 2 in coastal dune habitats and 2 coastal scrub habitats. Then we caught pollinators along transects for 30 minutes, processed and identified bees and flies to their species, and finally analyzed data using NMDS and Permanova. So this first figure is a non-metric multidimensional scaling plot, or NMDS, with a permutational analysis of variance value, or Permanova, of pollinator composition between habitats. For those of you who don't know for those of you who are unfamiliar with NMDS and Permanova, NMDS is an analysis approach that shows dissimilarity, and Permanova tests significance of differences. From these results, I found that there is no difference between habitats. Some factors influencing this result could be the distance between habitats, as they are within a mile range from each other, similar resources present, or community domination by pollinator specialists. Overall, these results suggest that pollinator composition is less driven by differences in plant communities between the dunes and the scrubs. This second figure is an NMDS and Permanova of pollinator communities between the past and present. Unlike our last graph, the 2 ellipses are overlapping less, representing a difference in community composition over time. A possible explanation to these results could be changes in plant species richness and or loss of pollinator species over time. While there is no difference between the habitats, there was a significant difference between the data from the 1990s and present day. The specifics about what caused this difference remains unclear because my analysis methods measure how different the 2 communities were and not what caused the difference. I can only claim that there has been a change in the last 30 years. However, I can provide some anecdotal evidence that suggests a loss of certain pollinators as a factor. During our survey period, we did not collect any pollinating flies from the genus Aristolis, but in the 1990s collection, 8 different species were represented. While these results are alarming, we can push for further research to what is causing the change in pollinator communities. It also leads us to question, how can we help our pollinators and be the change we want to see in the world? Before I end, I'd like to thank the CAMINO and Norris Center staff for being so supportive and organizing this program to provide opportunities for folks like me in research. I'd also like to thank my fellow interns for being the best group of pollinator pals I could ask for. Thank you.
Thank you so much.